Bismillahirrahmanirrahim. My name is Imran Khan and I am a student of MLT 8th semester. My registration number is MLT120192031, submitted to Dr. Niaz Muhammad, and my subject name is Seminar. Department MLT Cohort, University of Science and Technology Cohort. My presentation topic is dengue virus. The dengue fever was first described in 1780, and the dengue virus was isolated by Sabin in 1944. Dengue virus responsible for the dengue fever and dengue hemorrhagic fever. Dengue virus belong from flavivirus family. Flavivirus are spherical and 40 to 60 nanometer in diameter. Dengue virus is RNA virus and its outer surface is covered with envelope. Inside the envelope is a capsid shell that contains the virus RNA genome. There are two cell surface receptor molecules important in dengue infection. The cognate receptor involved in normal infection. The Fc receptor is involved in phenomena called antibody-dependent enhancement. This is the typical structure of dengue virus. This is the viral genome. This is the nucleocapsid. This is the ENM protein, and this is the viral envelope. Dengue fever. Dengue fever is a painful viral disease, also called bone break fever. Lead to severe flu-like illness. It infects 400 million people affected worldwide every year. Type of dengue viruses. The virus responsible for causing dengue is called dengue virus, or DNV in short. There are four types of DNV serotypes: DNV1, DNV2, DNV3, and DNV4, meaning that it is possible to be infected 4 times a single person. Transmission. How a dengue virus can transmit from one infected person to another. Dengue is transmitted by the bite of mosquito, Aedes aegypti. An infected mosquito from a human blood containing virus can later transmit that virus to a healthy people by biting them. This is the life cycle of dengue virus. Transmission continue. There is also evidence that an infected pregnant mother can transmit the dengue virus to her fetus. In rare events, dengue can be transmitted during organ transplantation or blood transfusion from infected donor. Epidemiology of dengue virus. Dengue virus are distributed worldwide in a tropical region where the Aedes vector exists or endemic areas. Dengue virus can commonly infect urban population. And this is the question about dengue. Can any type of mosquito carry dengue? The dengue virus is carried and spread by mosquito in the genus Aedes. Species in the genus Aedes. There are 3 species in the genus Aedes. Number 1, Aedes albopictus, Aedes palicensis, Aedes scotillaris. Incubation period of dengue virus. Incubation period of dengue is 5 to 7 days or 4 to 10 days. Sign and symptom of dengue virus. Symptom usually last for 2 to 7 days. High fever up to 104, severe headache, pain behind the eyes, muscle and joint pain, nausea, rash, swollen gland, and vomiting. That was the sign and symptom of a dengue patient. Diagnosis. How can we diagnose a dengue patient or a dengue disease? We can diagnose that specific disease through PCR NS1 antigen test by ICT, ELISA test for IgM and IgG. It is the explained form of the above. Which parameter of CBC affect in dengue? That is the question in CBC, that which parameter of CBC are affected. WBC will decrease below than 4,000. Platelets will start decreasing from 150,000. 1,50,000. Platelets will start decreasing from 1,50,000. Note, in severe dengue, RBC count will decrease also. That is the hematology analyzer picture of CBC patient. That is the hematology analyzer picture of dengue infected person. PCR test. PCR assay are sensitive. It means that PCR is a sensitive machine. Requires specialized equipment and training. For this person, require a specialized equipment and a trained person to run a PCR. Not always available in all medical facilities. It is not necessary that PCR will be present in every lab. NS1 antigen by ICT. NS1 stand for non NS1 antigen by ICT. NS1 stand for non-structural protein. It allows rapid detection on the first day of the fever. It means that we can detect the dengue infection on the first day through NS1 antigen by ICT. It means that it is very sensitive. It only take 20 minutes to determine the result. This is the picture of ICT for dengue NS1. A typical structure. ELISA, IgM or IgG. ELISA stands for enzyme link immunosorbent assay. IgM is indicative of recent DNV infection. IgG antibody are detectable equal or less than 1 week after infection. The presence of IgG is indicative of a past infection. Prevention. How can we prevent the dengue infection? Wear long sleeved shirts and long pants. Control mosquito inside and outside your home. Use mosquito nets and spray. Treatment. There is no specific treatment for the dengue fever. The best option to treat these symptoms are Nexam, esomeprazole 40 mg, and paracetamol. Maintenance of the patient body fluid volume by drinking fresh apple juice, sugarcane juice, pomegranate juice. Patient with dengue should seek medical advice upon the appearance of warning signs. Thank you for listening.